All right. Thank you, everyone, for coming, and also for the student online. So it's my great pleasure to introduce Dr. Daniel Robertson from Indiana Bioscience Institute. He currently serves as Vice President and also the Director of Data Science at IBRI. He was working as a IT administrator at Eli Lilly before joining IBRI. Officially in 2017 as one of the early administrators there, and he worked in Eli Lilly before then for over 15 years with various administrative jobs for research IT as a senior director for research IT, and also received numerous awards at Lilly and other industry sector companies. Okay, without further delay.
Great.
Thank you, then.
So thanks for the opportunity, and hopefully those online can hear well. I usually will wander. I'll try and stay closer to the mic so that carries. It's a great pleasure to be here. When I was at Eli Lilly, I actually filled in a couple times for the Dean's Advisory Council when this building was only just the graphics and everything like that, and still had not been constructed. So this is my first time in the building. Fantastic in terms of the little that I've seen at this point in time. In fact, past few times I've been down to Bloomington, have not really been in this area recently in terms of I've been at the Union and a few other areas around in the meeting, so I hadn't been here to see this, so fantastic to be here. It's also nice to sort of be back a little bit. If you looked at, and thanks for the introduction, because I really started my career here in Indiana at the IUPUI Department of Chemistry in there for basically, I think 7 years before I went to Eli Lilly. And actually, at one point in time, for a brief time, I was actually when they had the cave, and I don't know if you still have a cave or not, when they had the cave and the immersive desk, I was actually lead programmer. So there's still an image from long ago when I had more hair, when I actually was highlighting, doing some of the work, and I'd done some of the work with the virtual human and showing it on there. So I had a brief stint doing that, and that time I was actually coming down almost once a week to the Bloomington campus to do that. So there's multiple reasons why it's nice to be here. So from my time at Department of Chemistry, I was fortunate to join Eli Lilly. I spent 17 years there, and in my career I've always been, I think, having good opportunities. So now I'm with the Indiana Biosciences Research Institute, and my goal here is whenever I talk to people, I've got to talk a little bit about what IBRI is, what we're trying to do. And then for this audience, I want to go in a little bit more into one of our specific projects that we're going to do. So let me go ahead and move forward on this, and so we have more time because I've got quite a few slides and go through. But really, if you think about the life sciences companies that we have here, a stat that's not showing on my slides, but we really do have in exports, if you see the BioCrossroads reports, we are number 2 in life sciences exports. Why? Because we've got Eli Lilly, we've got Roche Diagnostics, we've got Cook Medical that's here, we've got Dow Agro, now Corteva, we've got Ortho, the Ortho Works up there in there. We've got a huge life sciences base. And we've got great academic institutions. But the recognition was in 2012, it doesn't seem like we still have as much going on between our life sciences companies and our academics. We are training, but we're maybe not retaining talent and keeping them locally. So how do we increase the opportunities to really retain talent, attract more funding, grow institution, grow a pipeline of talent, and really do applied research. So it was recognized that we could potentially do more. And so IBRI was created as a way to, can we take the great substrate we have here in the life sciences industry, in the academic institutions that we have here. And yes, I know that each of the academic institutions has a tech transfer office and their entrepreneurial activities, but can we IBRI facilitate bringing those together and doing things in a different way to really transform going forward? And so it comes back to, people ask, well then, okay, great, but we think we're doing part of that, or we're doing a different part. What does IBRI play? And we've really come back with a few different things in terms of how we try to operate within the ecosystem. And that is, we really want to be a catalyst. We want to pull people together, do things, and catalyze activities, but we may not be the ultimate owner of those. That will recycle from a chemistry. If you still remember that, a catalyst is something that accelerates the process but not consumed in the course of the process. So it's recycled to be used again to accelerate going forward. So we want to be a catalyst. We want to drive our things through collaboration. I'll show a mission, and it really is collaboration between the talent and the people we have within the organization. We don't want to compete with people, and we don't want to be redundant. So how do we complement the assets that we have, you know, within the region or even nationally as we go, and can we play together bigger? And I'll tell you some of the challenges in terms of doing that. And then sometimes as a component of that, convene people, play bigger, I sort of talked about, and sometimes it's connecting the linkages. We have people who have experience, we're out in the environment a lot, we're seeing a lot of things going on. Sometimes it's as simple as convening people, connecting people, and hopefully good things happen from that as we go forward. So a couple other things, when you talk about Biosciences Research Institute, that was pretty big, right? So then it came back and said, okay, so where are we going to focus? And the focus really came back to being, could we focus on cardiometabolic disease driven by nutrition, basically biology and otherwise? And when you think about that and the challenges that we have with the diabetes epidemic and otherwise, great. But even still, as I think of it, that's still pretty broad. So we have a relatively broad place that we're doing it. And how are we going to do it? It's really with a world-class organization of researchers, engineers, science, and business professionals that can really handle these type of activities, because it's not just about the science, but it's about organizational, it's about agreements, it's collaboration, a lot of other additional components or skill sets you need to drive this forward, and always with a mind of scientific excellence, entrepreneurial, because we're an applied research institute. So if you are applied, then what you deliver has to be consumed by someone to be valuable, either by the life sciences companies or as startup opportunities. We are a not-for-profit 501c3, so we are also independent. So although we have received funding from the state and from corporate partners and Lilly Endowment, we are independent in terms of this, and we have overarching MOUs in how do we operate with IU, Purdue, Notre Dame, Ball State. We have multiple research agreements with various life sciences companies in terms of that. So who we are from a leadership level and what does the Institute have? Basically, Rainer Fischer, most people hear about Max Planck, but they don't hear as much about Fraunhofer. So when you talk about or applied research institutes, the Fraunhofer is the largest conglomeration of various applied research institutes within Germany. And so he was recruited to join IBRI to try and do a part of that model here. Just this past year, now Jay McGill came in as our Chief Operating Officer after a long career at Eli Lilly in terms of that. So we've got a variety of senior leadership to help us going forward. And although we initially started with as a group of entrepreneurial-minded PIs who fit between or don't fit in industry or academic or somewhere we want to float between, we are now organizing ourselves into various centers. So I went on loan from Eli Lilly to assist, and I'll talk a little bit about that. IBRI Get Started. because of a recognition that really understanding data and data-driven and the digital revolution that we're in is critical, and many of the life sciences partners are struggling with how to do that. So I'm leading the Applied Data Sciences Center that we have. Another industry veteran, Mike Puja from Single Cell Analytics. He was at Siemens, Bayer and Siemens. He has joined and he's doing single cell type diagnostics and driving some of the things forward that he actually couldn't do in a business environment because they had different priorities. Most recently, just last year, it was announced the Lilly Diabetes Center of Excellence at IBRI, where jointly IU School of Medicine, Eli Lilly, and IBRI are recruiting talent within diabetes biology research. So Bob Considine from IU School of Medicine has part-time driving that, and I assume it'll transition. And then most recently, we had brought in Vedadi to grow a group in terms of biologics, vaccines, and antibodies in terms of that, because if you know anything about delivering value, sometimes your biologics or otherwise, when you understand what you're going after, is the fastest way to get something to market and validate hypotheses. So what you see is starting to grow in the Institute is really these various components that are very complementary, right, in terms of the diagnostic side, the data side, the disease biology understanding side, and then how do you translate that with a potential therapeutic or tool in terms of doing that. So we are on our way, but we're still relatively young, although I said 2012 was where we initially was conceived. It really was mid-2015 when the first CEO was brought on. So it's only been a couple years that we're growing. Now, one other thing that's taking place as we think about driving innovation within the ecosystem, where's the place to do that as well? Former CEO who's now with Purdue on innovation, David Broker, used to say that innovation is a collision sport, right? And so it's people coming together from different disciplines, different organizations to really drive things differently. And so if you are having a collision sport, how do you do it? How do you increase the probability of those interactions leading to something? And so as a part of this, although IBRI was originally thought to maybe live on the IU School of Medicine campus in Indianapolis, John Leckleiter basically said he had come from Cambridge, had seen what happened there, and said, hey, we've got this place northwest side of Indianapolis, downtown. Can we do something different? And so now, basically just the Northwest, just north of Regan Street, just north of the School of Medicine is planned to be 16 Tech, which is an innovation community, and where IBRI will be one of the anchor tenants. This is our building. We don't own the building. Developer owns it, multi-tenant. We'll have 3 of the floors. Actually, School of Medicine regenerative effort will have the 4th floor, and then the overarching thought leadership will be on the 5th floor of that, and that is CICP, the Central Indiana Corporate Partnership, the CEO group that has been driving economic development and these changes over 20+ years, BioCrossroads, TechPoint, Ascend Indiana, Ag Novus, OrthoWorks. These entities will actually be housed, moved from the Salesforce building down into the district to really drive this innovation in terms of there. Ground has broken, and just like this building, I think went up relatively probably quick compared to when I recall timing, or just I'm forgetting timelines. Time's going by maybe too fast. We have broken ground, and the plan is to be mid-2020. So let me take you on a little bit of a journey then, and then I'll focus on a use case in terms of how I on loan to Lilly, and then what Lilly called me back a year and a half ago, and with what we were doing to IBRI, I said thank you, and IBRI countered, and then I continued on with IBRI in this. And let me tell you sort of why, and I'll use some of my historical slides and then go into a little bit about one of the collaborations we're doing that as a more specific use case. And we can talk about a few more things as we need, and I'll leave time for questions. But really, this is one of the early slides we generated at IBRI. We'll probably change this because, like any organization, we put yourself in the center, right? We're always the center of our own world. We don't think we need to be the center of the world. If you think of a network-based, we're absolutely happy being at the interfaces of these, and we actually think we'll be in the interstitial spaces, helping organizations like these be here. But we are in the place, what I really wanted to show by this, and what we show is, we have huge global multinational companies, and you go around there and we have payers, and we have healthcare providers, and we have the research universities and the institutes, and a growing technology startup here in terms of their life sciences entrepreneurs coming from and hosted lots of times in the various tech parks and otherwise, and a very supportive business environment. And so it really was, when we started looking at this, when I was coming on to try and figure out how can IBRI be successful, what can we do in the space of this? How do we really exemplify IBRI to meet the aspirations that it has? And for me specifically, I was brought in because of big data. What could I do at IBRI in the space of big data? We all know it, right? We've got Google, we've got people who have their own data, we've got Apple, we've got all these other type things, tech companies and everything that are going to disrupt it. What place can we play within there? And so that's what I hope to show a little bit, at least the first hints of how I think we as an institute and we as a region can compete within that space. And so when I thought about the mission and everything of cardiometabolic health and the various stakeholders that IBRI had, I met with all of them. I was at Cook Medical, I'm at Lilly, had more historical knowledge of Lilly. I'm with Roche, I'm with Dow Agro. I'm meeting all of these people, asking them, what's your problems? What do you need? And it's all within their various scope of deliverables in terms of therapeutics or diagnostics and devices and nutrition and wellness programs, because we even have IU Health and some others. And so it became very clear that if we're taking a data-driven approach in terms of this, these are the outputs that we want to support in some way. And so by working backwards from there, and a data science is, okay, so it's about the data. And it's about the integration of the data and driving insights of the data and applying it to it. And so then I thought, what are the data sources we do? Yes, it's the research data that I had to live with in Lilly for a long time. So all of the chemistry and biology and genomics and all of that type of stuff, the clinical data, what happens in clinical practice? How do we understand the patient, the individual, the therapeutics, how the biology changes into a disease state that somehow has to be rectified. Now, when you're in cardiometabolic health, yes, the genomics part is maybe 20% of it, but it's our environment. Where do we live? It's the metabolomics, lipidomics, those other things that are influenced by our environment. And so it's also socioeconomic. And then you come back to, okay, with all the connected care and devices and otherwise, and our behavioral, there's a behavioral component. So could we do something within the space, looking across both the types of the data and the organizations we have, and do something different? This was the original slide that I used just after joining IBRI with the Scientific Advisory Board. I've changed it a little bit at this point in terms of where we are. The mission hasn't changed. How we're getting there maybe has changed a little bit. And but our real goal is, and I'll try and highlight this, is how do we, and we had just a little bit of pre-conversation around this, right? How do we integrate across these various data sets, the clinical, the socioeconomic, the deep biology, behavioral, digital? Everyone owns it, right, differently? Organizations own it differently. Was there an opportunity for IBRI as a not-for-profit, independent, to sit in between these organizations to do it? Now, I always think from one direction back, and maybe in a non-sequential, what's the problem we're trying to solve? Can we get access to the data we need? And then we can apply all the cool technologies. So I start from more of that way of thinking about it, as opposed to there because we're applied, as opposed to saying necessarily it's not our goal to evolve the new technology. Others are doing that and very good at doing that. When I joined, we were not creating data. We are now starting to create data in labs. So I didn't have access to data, but I could help try and identify problems in which we could lower the barriers and be the catalyst. So the problems we went after was, what are the obesity rates statewide? First project I did where we worked with IHI, getting data to look at that across, and I'll talk about that briefly. What I'll spend a little more time on in this one is, can we actually use the data we get in drug discovery or in discovery within basically ag chemical products to really understand toxicity of small molecules in the early stages of discovery. Can we better understand, this is probably my biggest project ongoing at the moment, can we better understand type 2 diabetes? Core to our mission, and the comorbidities that result. Type 2 diabetes, if controlled, is not the issue, right? It is all of the complications that come from lack of glucose control and people not taking their meds and everything, and how it goes to the cardiovascular, chronic kidney disease, the neuropathy, all of the other type macro and microvascular events that happen. One of the other ones is in certain areas, and this we stray a little bit further from our mission in terms of diabetes, but we are clear there with the data. We have a lot of things where we don't have good observational metrics in terms of what's happening at a patient level. It's all patient-reported survey data. Can we use the new wearables and connected devices to actually understand patients and what's happening with them before they really know what's happening themselves, and remove a lot of the other issues? And then in the context of type 2 diabetes talk, we just had a paper published, I'll highlight in a moment, on what's your probability predictive modeling. And so premise really, coming back to this, one of the key things is we've done a lot of projects, we've got some renewals, and this I use to highlight the complexity, right? We are operating in a very complex state, let alone thinking about two organizations like an IU and a Lilly, because I lived Lilly, coming together to do a collaboration, and how long those agreements may take in terms of 9 months or a year. Start thinking about moving it from a two-body problem to a many-body problem, and add legal in the mix, and all of a sudden it becomes very frustrating. We've been able to, as a small independent, actually break through some of those barriers, take some risks, do some things, build some master agreements that allow us to move faster and focus on the problem. But it didn't come without a lot of legal agreements, data agreements. So that's when you're talking about, okay, great, couple projects. It translates into a huge amount of legal protection, security, and otherwise in terms going. So we do think that we are not independently in terms of doing this, but we can be a place to break down the barriers and really drive innovation in a different way, starting with the regional capabilities we have, and ultimately going more globally or nationally and globally, because we do want Indiana to be the place to do research. One of the best place research aligned to our mission, because we have lots of our partners in diabetes, diabetes care and understanding, as well as Diabetes Research Center, School of Medicine. So specifically within diabetes, we think we can gain more national recognition as a whole, as opposed to just independently. So let me go quickly through a couple of those projects. So I talked about this a little bit. This was my first project that I got done. I had the concepts and I had to make it real. So it was actually talking with, Darshan Shah's name came up. Prior to Darshan, basically was David Matsusoff, who was the director of the Management Performance Hub. This is where they're integrating all the state level agencies. Talking with him, we're trying to figure out, is there an opportunity to do a collaboration? The kicker came when we talked with Dr. Jen Walthall. At that point was Associate Commissioner at the State Department of Health, and we met with her, and the state said we've got 3 KPIs we're trying to do. One, smoking cessation. One of them is infant mortality. One of them is obesity. We've got programs and understand what we're doing in 2 of the 3. We don't have anything in obesity. I said, great, that's aligned with our mission. Let's see what we can do. Now there is an additional one, unfortunately, in terms of opioid addiction. That is KPI to change. So we went and said, well, let's see if we can get access to the data. So went after multiple parties, ultimately landed with IU Health and iHi, Regenstrief's operational arm, or however you want to say they're independent, but they operate off the same database systems. And we pulled the data together. We pulled it together with what they had at the MPH at state level, and we generate some dashboards of obesity rates across the state. And then we compared them against CDC survey data as sort of the gold standard of what those looked like. What was really interesting, I had people say, why are you going after EMR from IU Health? Because, you know, iHi has all the data. No, they don't. Actually, when you look at these types of data, we were going after BMIs, heights, weights, and everything. Actually, they weren't the way iHi was initiated in terms of exchange of data. That data primarily sat in EMRs. So even though iHi and Regenstrief had 14 million patients in there of something, their prevalence rate of BMI data was less than 5%. IU Health, much better, although they only had 1.5 million, right? Its prevalence rate was actually over 50%. Why? Because every time you went in there, they took your height and weight and they generate that, and it typically. So based upon that, there was a couple things that was done. One, immediately, iHi now, for supporting public health and otherwise, was coming back and now starting to work with their healthcare providers, transfer that data into our data set so we can start doing it. It also informs some of the state level in terms of how we need to better integrate our data sets to drive some of these population health and public health efforts. We are now in conversations. We were continuing to drive this path and then get disrupted a little bit by Jen Walthall moving to another organization, David Matsusoff becoming a consultant, Darshan coming in, and now we're back in working with entities to try and understand what can we do this year in terms of this, including IU Bloomington School of Public Health, who has a effort in as part of the CTSI grant in terms of rural obesity. So stay tuned, and I hope I can take this from what was a now aging to something new that we're doing in the space. But that really led us to what is one of my bigger projects with is the health records. Working with Regenstrief and coming back originally with Eli Lilly because they want to understand disease understanding and biology. And working with them when I showed it at Scientific Advisory Board, Roche Diagnostics said, hey, we're doing something similar. We want to understand data. Can we jump in and help? And I said, absolutely. And so we did an agreement with Eli Lilly, Roche, IBRI, pull in Regenstrief, IU University, Notre Dame in terms there. This has now been ongoing for multiple years where we are taking the relatively raw, messy, hard to analyze EHR data. We have a pipeline to, and most of the work has not been on diagnosis codes or procedures or some of those standardized ways, but even when you have link codes for observations and clinical variables, very messy data. In terms of how many fields that gets in and how diverse it is by the different methodologies. So we spent a couple years now using modern technologies, cloud-based, PySpark, all the other type things, mappings and references to really generate this really massive 20-year data set over a terabyte into a research quality data set that can be analyzed in terms of this. And we actually have people from IU School of Medicine coming to us to say, can we access your clean data set, because if I get the raw EHR data set, I don't want to spend the 80% of my time or my postdoc's time trying to clean that up. So this is ongoing. We're continuing to do that and working with Regenstrief. In fact, as part of this, I'm now an affiliated research scientist with Regenstrief, where we're doing some of these efforts. And most recently, as of Monday, there was a publication in Nature Medicine from this collaborative effort where Roche Diabetes, in conjunction with IBM Watson and the Explorers data set, generated a predictive model that basically said, can we predict people their probability of getting chronic kidney disease within 3 years of first type 2 diabetes diagnosis. Where we came in is they generate the model, they did the standard cross-validation of the model within their data sets, but we provided a similar data set from Clean, from INPC with Regenstrief, so that they could validate that. So we became a part of that in terms of validation. Actually, for a year, that was under review and some changes and revisions going on, because the editors wanted and the reviewers wanted a few more things done, and those additional random forest models and some other things were delivered in terms there. So have a look at that. One of the key outcomes of this is lots of times we work in a very clean data space. So let me say clinical trials. They're very well defined, they're very clean, you're getting the same data on everyone. The moment you go into real world data, very different. And so the premise is, if we really want to do things that operate in the real world, let's use the real world data to do it. Because it makes no sense having predictive models or AI that's based upon things that you actually can't compute in the real world. So let me move on, and I'll move relatively quickly in terms of here. I'll come back to this, human toxicity, and then I won't talk too much about this, except that we do have our own, how can you live without your own app? So we do work with small tech company. We have our own app that is using Apple's iOS HealthKit, CareKit, and otherwise to collect data directly from patients. It is in use at School of Medicine, Stanford Health. It's part of a CTSI RFA. It was finalist in the Inject Tech Challenge, the inaugural one that was done in October, and we are potentially launching this as a startup shortly here to drive revenue and growth with that, and I will continue to use it for research purposes. So just coming back to hit this, so when I went into this strategically to start thinking about it, I thought about organization and I thought about data. And so if you look at the projects I just talked about quickly, we started going across IHI and otherwise in the geo-encoded obesity data, which actually is a limited data set, which taught me a few more things as I went. Type 2 patient understanding, we started bringing in more people into bear. If we're going collaborative toxicogenomics, I'll talk about that. That's more the biology, deep biology side, RNA-seq, Affymetrix, gene expression, and then digital biomarkers and digital behavioral. And oh yeah, by the way, there's a Fairbanks Institute that is now since closed, where we actually had the EMR data and the deep biology data that we're working with for analyzing. Unfortunately, too small of a subset to do have real statistical power in terms of. So let me now focus for the last about 10 minutes or so and leave time for questions in terms of collaborative toxicogenomics project. Before I do that, let me just come back and say, highlight a few of the things in here, and I didn't know where to put this slide, but I thought it was good to put it at this point because we are a small institute. In fact, much of the work that I had done up to this point was done by acting as virtual teams. We're real people doing real work, but the team was relatively virtual. In terms of that, we used consultants, we used in kind. When we're working with Lilly and Roche, the Lilly team usually is every Friday at IBRI working side by side with us. So we are doing these things to act well beyond our size. But at this point we have, and Brad's in here, and so he'll he's joined me today and will be in part of conversations ongoing. He's now driving the project I'm going to talk about, so he's probably the expert who should be up here talking about this. But I actually stole someone, or recruited away someone, maybe a better term, from the School of Informatics and Computing, Meeta Pradhan Rangki. Her husband had actually joined IU School of Medicine. She's a PhD biologist who got frustrated with doing lab-based work and went back and got her master's of computer science, and so she's in the lab. Brad is a long-term veteran of Eli Lilly who took the early retirement package, and I was able to recruit him to come here. And then also we have someone who just went through the accelerated Notre Dame Esteem program for MBA, Keen O'Donnell, who's actually working with us on the digital biomarkers. So those are great. Those are some of our internal staff, but we want to operate much bigger than our size. And so I have to add in the key collaborators. Most of what we're doing, you'll see another slide of acknowledgements. Huge number of people that we have working with us in this. So this comes back to us saying we're convening people, we're collaborating, we're doing that. We don't have to own everything ourselves. We just have to bring the best and brightest together, focus them on projects, lower the barriers for them to do good work and drive forward. So let me talk a little bit about the CTOX collaboration. It took a couple different directions where we were. But first, why is it important? If you look at drug discovery, I grabbed this publicly from Lilly. The drug discovery process is very challenging. In my, well, as an individual contributor for 3 or 4 years, any project I worked on individually, helping in drug discovery, where kinases, oncology at that point, failed. None of them progressed in there. They did inform other projects and everything, but there's huge attrition rate as we do drug discovery. That leads to the high cost of therapeutics in terms of this. And there's critical components in there where animal models, either for safety or for disease understanding, are used within there to drive the process. And many times they don't translate to the human experience. If you look at Corteva, Dow Agro, they are doing crop protection type of chemicals. They tend to be very different than what you do as pharmaceuticals because you actually don't want them to go in the human body and cause effects. So very complementary, but their process is very similar. They're just optimizing in different ways. And what's really interesting is they do almost the exact same studies for EPA in terms of understanding safety of these compounds. So if you start looking at them, both of them have to understand this. And there's a real effort, and like with many things, there's hype cycles and everything. The whole thing was transcriptomic. Can we understand the gene expression RNA sequencing profile in terms of that? And both of them started moving similar directions in terms of this. And oh, by the way, I always thought, oh yeah, ag, that would be a really good business to work in, right? You could get your products out fast. No, it's about the same timeline as getting drug on the market. If you look at that 11, much longer than you know, many of the other things that are up there in terms of there. So these discovery processes are very challenging, and we do think that better understanding of the data, better understanding of the studies can actually drive that. And especially when you talk about animal studies, there's a whole effort to reduce them and remove them when you can and not use them going forward. So other key thing is with the life sciences coming, especially Lilly and Dow Agro, they had similar genesis in terms of, and Brad, I think, was there from the start in terms of some of this with Dow Agro and Elanco, and otherwise I should have had them up there. Those are the three organizations. Now Elanco is independent from Lilly, where people would sort of discovery scientists would move between those organizations. In 2015 came recognized we're doing a lot of similar things. Can we actually explore what we could do together? So the discussions had started. We really came back to we're working with compounds, and we have these safety models that we have to. So it came cheminformatics and toxicogenomics. I had to excuse myself. That's when I went on loan. I got firewalled from Eli Lilly because there's now IBRI that actually became a benefit, because then IBRI became a place where we hosted these collaborations. We explored these, and then in a corporate environment, a restructuring happened. Dow-DuPont merger, changes continue to go on with Lilly. That's one of the challenges working with industry partners, is there's always the internal change that's ongoing. But ultimately in 2016, we did get alignment that toxicogenomics was the opportunity. It was a collaboration of equals. Both Dow and Lilly felt that they had very strong IP and knowledge in there, but they were operating in different markets, and so it could be there. In the cheminformatics and predictive modeling, Lilly thought they were ways ahead, and that Dow would get more benefit from that collaboration than Lilly would, and so it was not of equals. So we did do this as a 9-month pilot to generate this, research completed, and we were moving on. So what were we trying to do with collaboration? Originally, I thought it was sharing data in a secure way and let's generate models across there. What actually came back to was basically Lilly and Dow said, you know, we're not able to talk as much about what we're doing and how we are making industrial-based decisions. Regulatory scientists are moving slower than we want them to do. We're doing experiments we don't think we have to do if we use the data right. And so can we use IBRI as a not-for-profit independent research institute to actually influence regulators, scientists, and academic colleagues to move the whole field faster? So instead of it being us helping the industry, it was us helping the industry move it back out into a broader way. So it really was to deliver a shared platform to deliver these harmonized analyses, bioinformatics analysis. It really was to expose the decision-making. It was to use some of the more network-based approaches rather than looking at single gene expressions or otherwise, or compound similarity, and move it to really a more biologically relevant discussion of what is happening within there. And then from a technology, could we deliver the shared platform that ultimately would go into the open source? Could we build it from open source tools, contribute to that, and lower the barrier of delivering it to this? The data we were working on was transcriptomic type data. Old style was the AFI chips or comparable gene expression chips. Now we're moving with the advent of the sequencing into people doing a lot of RNA sequencing in terms of this. There were publicly available data sources available, both GEO, which is still going, and then a couple other efforts of drug matrix that got acquired and was made to the public, and TG Gates, which was done in Japan to try and understand what happens when you dose animals with these compounds. The methodologies we use, Lilly uses a published workflow 2 years ago now, in terms of this, where they're doing the weighted gene co-expression network analysis. So rather than looking at single genes, can you actually look at genes that co-express together on average? Can you then put them into these dendrogram networks there? And they actually start understanding and showing the biology of what's happening. And why? Because there's a lot of things within our human biology that we don't understand. There are pathways that are not curated. There are pathways that are not understood. So if you only go a curated analysis, you actually are missing some of those type things. Additionally, for the experts looking at this, they actually know which biological processes are conserved from rodent to human, and then you could start making these assumptions saying, if it is hitting this, okay, great, we understand that's rodent biology. It's not what we expect to see in human biology, and you can make rationales. Corteva moved the same way, using a different type of graph-based approaches, more of a arachne-based approach, where they likewise put these gene expression results into modules to really assist in their understanding. So to cut to here, and we absolutely will not have time for a demo, is we generated a publicly available platform. It is available here on there, ctox.indianabiosciences.org. Available there. We are hosting multiple instances of that at IBRI for people and demoing it. So if anyone's interested, we're available to do a demo at some point. Just reach out to me and we'll get some scheduled, and I'll probably give it to Brad quickly to do the follow-up in terms of this. But we've actually set it up where we can actually generate and take the results of new studies. With another pharma company, not one of the collaborations, we've just done that, where we've actually taken results that they've got, run it through ours, help them interpret the results coming back and deliver the report back to them. So we're seeing a piece of that. So you can upload your studies of the results with the controls and the dosing and the experimental study design. You can analyze, compare, you can visualize the results and assess the risk. So yes, we do standard expression data, and looking at this in terms of here, these are really showing either your proprietary, or we've analyzed everything we could within the liver from TG gates and drug matrix, and this is what you're seeing here. So not only if you do your own one, you can look at it in comparison of everything else, all the other experiments that have been done. So you can go through and do these analyses and quickly, they're pre-analyzed. You can then start looking at gene level analysis, the whole genome, wow, I just changed on that. The weighted gene expression or co-expression networks, or these GSA type ones, or find similar experiments in the database. And if you start seeing some of these things, actually look at what has been annotated and curated in terms of histology in terms of there, and view them a variety of different ways. So looking at gene level results, you can basically pull them up, analyze them. They've got all the p-values, the normalized, they're showing you the genes. You can link out to what the genes are. You can understand in this clofibrate example, you can actually see that there are some p-power and cell cycle pathways that are being hit. You can go a little further into the co-expression networks, look at them in terms of bar charts, put them back on what Lily had defined as the diagram. Now, what I will say is, analyzed them from the diagram, a little bit difficult, so starting to do some annotation in terms of that, as you see in the red highlights, so you can start understanding traditionally, if you're hitting these type of pathways, what do they lead to in terms of toxicological results. So there is a database contextualization that happens in the course of this. Gene set analysis, so you can go against your favorite curated gene sets and goes and otherwise, and start understanding those in terms of there. We've just done a new way of analyzing this that is the publication has just been sent out to go back and say, can we start reducing these large sets down to representative? Think of it as cluster-based or co-expression network, in a different way of what Lily was doing with weighted gene co-expression network. But again, to try and get more rapidly to understand what is happening within the biology for an expert toxicologist, so that they can understand the next experiments to do driving this forward. So you have a variety of different ways you can look at those. I'm going to do this. You can do some similarity analysis. This is not from compound similarity, but similarity of expression. You can look at what those are. And again, as I said, historical histopath, clinical results, and even some ways to start predicting some things. And when we talk about prediction, you usually do short-term studies and long-term studies. So this is coming back and saying, can you actually, within the first couple days of dosing, can you actually predict what you might be seeing in a 25-day study, right? Can you see the hints of toxicology coming really early that you then need to understand what you need to be looking for in there? Or can you tell that it looks like it's relatively clean in terms of that? So originally, if I had more time, I'd do a demo. So let me finish up in terms of this. Where we're going next is really compiling user requests. We are making this open source. It's publicly available now. We've limited demos and people to accounts until you do a demo and do this. We're building a user community both in the ag, chemical, in the pharmaceutical, and in the academic environment as well. We are looking for additional funding mechanisms, continue this project, and in a literal sense, where are we going next? We will be exhibiting. I had the opportunity to do that last year, and we'll do it again this year at Society of Tox on the exhibit floor, talking with various people going forward. So I hope to what I have sort of shown you up to this point is we really are trying to live in IBRI space of how do we catalyze, collaborate, complement, convene, and connect the right people together to really drive problems of interest in cardiometabolic health. We are doing it by dealing with the data, wrangling it, cleaning it, integrating it, and we are doing some things where rather than just looking at what's available to data, hopefully this year we'll actually be going after specific data creation of patient samples, of cohorts of interest, ad hoc cohorts of interest. We're acting as a collaboration hub. We don't have to do everything ourselves. We bring the right people together, provide them the tools and capabilities. We do have to be expert at trying to understand what our life sciences companies are trying to tell us, what the environment's trying to tell us of what are the problems of interest, and then applying that. And we are taking a very, because we are applied, and we're expected to deliver on money that is given to us or to us, we take a very delivery project managed approach in terms of where we're doing. And so I'll end on this slide, which is, it was not done by me alone. I sometimes tend to be the face of this, and I'm sure I'm missing many people who were here, but what I'm showing you now is really a much broader integration of a lot of ideas from a lot of different people, contributions in time, data, in a lot of those to really do some, what I think is really interesting, challenging things as a startup and with relatively low cost in some respects in terms of what we're doing. Now that continues to be a challenge because we are not for profit, but that means we still need to cover our costs at some point. So we are continuing to try and do that. So stop there and questions.
Thank you very much, Dan. We have time. That's some questions from the audience and we can go to
right. Yeah.
Quick one.
Sure.
Drugs don't usually fail clinical trials. They usually fail because they don't want.
That is actually true now, as opposed to it was 15 years ago.
Yeah, 15 years
yeah.
ago, it was roughly 50,
Correct.
50
Correct.
toxins.
Correct. And so,
now it's all moved over,
so,
they fail rapidly.
so you're absolutely right, and that is a key piece. But a part of it is because of this work that has been done internally in industry to try and weed out some of those things and reduce and coming back to. And so let me follow up on that. So this is the toxicology, the safety. Actually, they get screened out early, but you're absolutely right that what we have seen in the drug discovery, there's a couple reasons that I think in terms of failing. They usually are now failing, right, for lack of efficacy in phase 2 and phase 3. And it really comes back to, I think, my bias in terms of this is one of them is we've gone off after a single target hypothesis. That somehow we think that we now understand a biological pathway, we've identified a target that appears to be associated with the disease. We are a very redundant, robust environment. And so what happens is we may get unique and very selective blocking of that target. Unfortunately, our body has lots of different ways to cascade around it. So single target hypothesis is it. I do also think that actually some of it is that we're over-optimizing in disease models, animals or otherwise, that really don't translate to the human experience and in the environment in which we live. And those are just two of the many reasons why it is very challenging. But you're absolutely right. Over the course of my time at Lilly, we moved a lot. Quick anecdote on that. Because I spent my time in discovery chemistry in my early part of my career, then I moved in 2010 to the research IT. I was supporting the informatics group. They came back and said, hey, we've got a PGP pump inhibitor problem that's causing us an issue in terms of getting this compound here. I'm like, what do you mean? We've understood that in discovery for the last 5 years. We have predictive models to optimize away from those properties within a compound. It was like looking at the sun, right? Or the stars, right? What I was looking at was a project that was never informed by that point of time and what we knew at that point in time. And so now they're having to resolve those issues later stage. And so that came back to me that not only that, but the timeline of drug discovery into clinical and everything, you're looking back in history. You're looking back at the science we knew 5 to 7 years ago, not what's current.
Well, it's clear that
So,
the omics revolution has not led to a medical revolution.
correct.
It just hasn't happened.
I did chemogenomics. I'm still amazed that when I started at Lilly, we didn't know how many kinases there were, right? and somehow we thought that, and how much are druggable and those, and so no, we are naive.
Another
So.
issue is, so we know in pharmaceutical failure to do toxicity is not
Right.
But that raises the question, is that because we're being overzealous and not taking compounds because of some toxicity? In other words, the way you don't get toxic drugs is if the rat sneezes,
Right.
stop the drug.
Yeah.
Anything else with it?
Yeah, there is a piece of that. I think there is a piece that it's a very risk-averse environment, and so you may be overly conservative in terms of what you're seeing in the process for planning for the first human dose. I absolutely agree. We still get surprised, right? The good thing is that now, you know, it's been pretty good in terms of not having adverse effects in first human dose. But I do think that piece of that is we're overly conservative maybe in something that might not translate, might not actually be a issue in humans, but no one wants to take that risk from a business environment. Huge liability.
Funny too. A patient's perspective and appetite for cost benefit and risk benefit may be completely different than a toxicologist.
Yes.
Saying if you're living with a disease for 30 years,
right,
you may be willing to put up with bad
Yeah.
effects that on paper you say let's just leave it enough. Sorry, let's not even go to phase 1.
right. So actually it's interesting because I do see the risk benefit curve if you in oncology space or otherwise. You can actually have a much higher risk profile. When you start going into the chronic diseases like type 2 diabetes and those, they actually are more conservative lots of times because you have longer times for those to on which you'll be on those therapies in terms of that, where those adverse events can accumulate maybe over time through a mechanism that you can't see on a short-term study. So that is, we've seen those things, right, that have shown up. And additionally, in the drug discovery process, you are in a very small patient population, and sometimes it's not an optimized patient population for the people who may actually be effective. I think that is one of the other reasons is your hypothesis translates to 20% of your population in which you've done the clinical trial. At that point, you've just, you know, people are trying to figure out that post facto as opposed to trying to do that. We're getting better maybe in terms of that. Let me just state I don't know. I mean, it's huge challenges. I don't think the industry knows how to solve these. I do think they will be solved by innovators or otherwise coming up with different ways to do it. Yeah.
One other question. That was more it actually happened from more of a cancer side where there's much more appetite and then patient advocates are also pushing things. Hey, don't block these things.
Give me access because if it helps me, I need it, right? From an individual base, yes.
The other question too is, you know, a lot of the omics stuff is single time point big data analysis. It's very, you know, shallow, but lots of it, and not a lot of time course information. Is there interest in and room for, you know, dynamic systems modeling and just dynamic analyses, you know, systems type stuff, but not just big networks, but the time dynamics?
So, absolutely. I lived through a time, I think there's always a silver bullet, right? You're looking for the right thing. I lived through natural products, combinatorial chemistry, high throughput screening, give you a favorite buzzword, and all of them, systems biology was one of those, where it spectacularly failed, was oversold and failed at Eli Lilly because in the end, all it showed was either things that were obviously right or obviously wrong to the expert. It didn't actually help you generate the hypotheses that were testable that would drive research. So I do think at that point in time and with what we have and what we have now with both compute power, what you're seeing with graph-based, variety of things, even better data coming back, we are moving into an era where those type of things and complexity of systems and modeling those simulations, my background is in simulations from what I did. I was surprised we never did much simulations at Lilly. It was all data parallel type activities we are. I do absolutely think that in the right context, rather than the static views, the dynamic views and simulations are a opportunity. Yeah. Yep. Yeah. Yes.
So for the agrochemicals, I wonder if you work together with EPA and NIH, the National Toxicology Program.
Yes.
Because many of the compounds are the same if you're using
Yes.
their resources.
Yeah, so absolutely, and we're in conversation. Brad can probably is more deep in these conversations, so we can do a follow-up in that. But that was a key piece of this. We had conversation at SOT with the regulators, regulatory scientists, with some of the organizations that are doing that. You're absolutely right. These were comments we just had this past week in terms of with one of the other ag chemical is that we optimize different ways in a pharmaceutical biologically active bioactive molecule that we want. We look for different characteristics. By definition, we weed out a lot of compounds that we'll never take into animal testing. In the ag chem, down Corteva and otherwise, they actually optimize compounds the other way, but still do similar type testing? Absolutely. I think there's a complementarity that could be very valuable in a more broader understanding as opposed to just being pharma focused. And that's where we come back to, even with benchmark dose calculations that are needed by EPA, that is being, it's in testing now where we use that. We are going to continue to expand and provide additional tools in context across the two different, at least two, maybe more, but that's our target markets at the moment. Yep. Yeah.
More from, I do a lot of work with the undergraduate population
Right.
here, and so certainly one vested interest we have is there better ways we can develop relationships between our undergraduate students and internships with companies? Sometimes it's hard for them even to get in the door and build these relationships.
Correct.
In terms of industrial work, like Claudia's working on.
Yes.
Just more general mentoring.
Yeah, I was just at Ball State before the end of the year, same type question. Just at Butler, we're working with them on the pharmaceutical side in terms of giving the experiential, how do you work with industrial partners, how do you do it in there. We are working along those lines. We've done a couple interns, we're looking at programs. I know the School of Informatics, I just met with the guy yesterday, Gary Shellback, who is starting a center there to do the same type of thing. You know, we've hosted graduate students, we've done those. So the answer is yes, how it exhibits. What I want to think about doing, and with the various people asking that, is can we do it in a systemic way, right? A program that actually can span beyond just IU Bloomington or IUPUI or otherwise, because I actually think there's value not only in the you know, in the industry, experience with industry. A lot of the efforts you do in industrial setting is team-based, it's cross-disciplinary, it's cross-organizational, and actually starting to do some of these things where you can actually have students and talent from different schools working together gives them different things as well. And then the flip side of that, I just worry, I lived in industry. I saw everyone coming at us saying, we'd like to do this, this, and this. Very difficult when you're focused on your projects, your work, and everything else. It usually takes a one individual to champion that at high effort. We want to lower the barrier in terms of doing that, where we can host people, we can do it, we can make it where the people can come on site. Even if you go to work an intern at Lilly, your first probably 2 weeks, if it's in the lab, is doing safety training or other type of training that you may not need to do in a different environment where you've access to the same data, same tools.
Onboarding and hearing council.
Yeah.
Because right now they're like, whose door do I go knock on?
Yeah.
And if you're doing it 10 times with
So,
10 students, it's really
so keep that thread going, because I'll be having similar conversations with Ball State, with other ones, and we'll be looking at how do we do something that multiple parties from an industrial side, but also from an academic side, can act as a venue for those type of experiences. How much can we provide? That becomes a challenge. So the other thing we're talking about is how can we actually make, for data science groups or otherwise, some of these data sets available So they're more real world, so you can do more real world things, but in a pseudo controlled environment, as opposed to, I just had an intern from very talented machine learning. When I gave him some messy data, he made all the wrong mistakes because he's used to getting clean data sets. So can we teach them that life is not always, here's a clean data set, get this result, run this algorithm on it. How do we teach them more in that? And that I think we can do more systemically by providing them with better experiences, sort of more real world style data sets and otherwise. So both those angles are viable. And so over time, we need to continue to do it, because the one thing that keeps coming back from our industrial partners, from when Amazon HQ2 was looking here, do we have the talent pipeline? And that's what we need to create.
I also have a question about the internship opportunity.
Yes.
For undergraduates.
Yeah.
Any business groups that I can go for education for my undergraduate?
Yes, and so, well, what's happening is where I'm trying to think of where we are at this point. From an IBRI side, I think we've pretty much defined who we will be having as interns this summer. We can only take a few because we're still small. We're only 40 people. But as I was thinking to Lily and otherwise, lots of times this is the timeline or past the timeline somewhat in terms of the internship program. So usually for summer interns, it usually means that most of those things are being done in the fall, which seems really far ahead. We are thinking about, in the context of this, building up the capabilities where it just doesn't become a summer thing. It becomes co-work, it becomes coming in. Up to this point in time, because I've had to move fast on delivering value to the projects, I've had to default to consultants or staff or in-kind resources, have had some opportunity to help with graduate students or otherwise. As we go forward, the other venue we're coming back to is, can we actually start setting up opportunities within semester for these type individuals to work on critical components of key projects as part of a team. So I don't have a clean answer, sorry for you on that, but I'm willing to grab my card right after and keep in contact. So
Damage and tech point is one of the future admins of this morning to. That's what we use for the summer internship coming up. Absolutely 90 students in this program. We're actually pretty close.
Yep.
So we're like step out, but they are starting this summer on this semester internship program. Exactly that.
Right. Yeah, and so this is where it comes back to is we think from across the state, right? I use central Indiana. We do think state. Then we think about crossing the borders of the state. So we do think about sort of broader regional initiatives. So, yep.
Okay,
Great.
I have a question
Sure.
about the toxicity testing. So your systems focus on the transformatics.
Right.
Do you use other omics data in case that the transformatics is too subtle to detect the toxicity?
So we have not yet. At Lilly, we absolutely were looking at a full omics type analysis across there, because there are certain things you need to do in terms of basically your genomics, transcriptomics, basically how does it translate into the proteomics, metabolomics, lipidomics. So add all of those together, because that's a key piece of it. You don't know what part of the process is being dysregulated or impacted in terms there. So we had efforts ongoing in the data side at Lilly to really look at it holistically across there. We are not there yet at IBRI, but have had conversations, and as the other centers develop, I've been more externally facing this year. In the next couple of years, I will be more internally facing as well with my team to say, how do we support them? But we will not support them only independently. We'll be coming back out and saying, okay, how do we get access to the right experts, right systems, right tools to help them do it? Not that I can never create the size of a group that I had at Eli Lilly. And that is not my intent and goal, right? From a tech tech side, from an informatics side, otherwise. My team has to be really, how do we coordinate these efforts? How do we know the experts? How do we help commercialize these tools, put them to commercial use, applied use, otherwise not really commercial, but applied use. So that is the opportunity as we go forward in the next 5 years as we develop, is how do we build this broader network of capabilities, funnel both talent, professors and otherwise, to leverage for these type of projects.
Wonderful.
Yep.
Thank you.
Yep.
Okay, thank you very much for coming and thanks
Great.
for
Thank you.